So the triglycerides, or triacylglycerols, if you want to call them that, are basically the main, probably the largest group of lipids you're going to see in living organisms. But there are other important lipids or lipid classes that you also see, and one really important type are the phospholipids. So the phospholipids are those little molecules that make up the cell membrane. They have some other functions also, but the cell membrane is their main role, and they have actually quite a bit in common with triglyceride. They are pretty similar. They are basically a triglyceride with the glycerol molecule here. On this three-dimensional model, the glycerol would be right here. It's a little harder to see, but it's this right here. And then a phosphate stuck to one of the carbons on the outside right here. And that's instead of the fatty acid that used to be attached here. And then what you have is the two fatty acids, two of the three of the triglycerides still over here. All right, so the fatty acids would be here on the 3D model. The phosphate would be here. And then this sometimes, a lot of times actually, phospholipids have another structure. They have another molecule, an attachment on top of the phosphate. So that it has a little choline hat here. You don't need to worry about what choline is, but there's a variety of different compounds that can sometimes stick on top. Choline is a common one. There's other ones that'll be stuck to the phosphate like that. So this kind of a structure makes the phospholipid different on both ends. So it makes it polar over here. So the head part of it, the phosphate head, is polar. It's got slight charges, or partial charges, I should say. Well, actually, let me take that back. These are full charges, okay? Like it has O minuses, so fully blown charges over here, like that. The point of that is that's hydrophilic. It's hydrophilic, and then the fatty acid tails, as you know already, are hydrophobic. All right, so when the phospholipids arrange themselves in a membrane, what they're going to do is the phosphate heads are going to stick out where the water is, so on the outside of the cell and on the inside of the cell, you're going to see the phosphate heads, and then the fatty acid tails reach into the middle of this bilayer. So they arrange themselves into a bilayer where all the fatty acids, the hydrophobic tails point in, and then the hydrophilic phosphate heads point out. And that's what you're going to end up seeing. Now, this is not necessarily a realistic depiction of the membrane. Yes, the orientation of the phospholipids is correct, but there's some stuff missing. There's cell membranes in living cells have a lot more other molecules floating in there. There's a bunch of membrane proteins. Sometimes there's carbohydrates attached to the phospholipids, and there's also cholesterol molecules floating along inside the membrane here. All right, let's actually draw a phospholipid. Let's draw one so we can get used to that and learn that structure a little bit better. All right, if we're going to draw one from scratch, the way we would approach that is by just simply drawing a glycerol first like this. Now I'm not going to draw the whole glycerol because I'm not going to draw the OHs because they've been involved in dehydration synthesis to link to those fatty acids now. But I'm going to draw it in this orientation at 90 degrees to what I normally would draw. You can draw it the other way too, but it's just easier to visualize this way. All right, I'm going to put two fatty acids here. Got that ester bond here. I'm not going to draw all the hydrogens in because I'm too lazy, but it would look something like that. You can draw the chains kinked or straight. These would all be hydrogens here. Okay, another fatty acid here. All right, and then over here I'm just going to put a hydrogen, another one, and then I'm going to attach the phosphate. So I'm not going to attach any choline or anything like that on here. I'm just going to put the oxygens. So one of them is a double bond O, and the other two have a negative charge on them. So this is where I misspoke before when I said polar, but these are charged, fully charged, which makes them very hydrophilic. It loves to be around water because those charges are like to associate with each other. So the tails then, this component of it, is then really hydrophobic. Or we could say non-polar. All right, so that's how we would draw a phospholipid. So practice that on your own. Take a blank sheet of paper or page in your notebook and just practice drawing triglyceride and then practice drawing phospholipids. Once you know triglycerides, these are pretty easy. You just have to remember that phosphate going on the other side there instead of one of the fatty acids. All right, another class of lipids that we're going to see in living organisms are the sterols, and they are based on cholesterol. Cholesterol is the main one, and there's other ones that are variations of it. And cholesterol has a structure that we're not going to memorize. We're just going to remember that it's these 4 rings. It's got 4 carbon rings, and then it has a carbon chain coming off of those rings over here. The detailed structure we don't need to worry about. The other thing to look at is that it has this OH over here, which is polar, has partial charges on it. All right, so cholesterol has a number of different functions. It is a precursor to steroids. So it gets made into steroids like estrogen and testosterone. So it's necessary for that. So that you might find that surprising that cholesterol actually has an important function like that in the body. So it's not just a harmful thing for us, it is actually also a useful molecule. The issue with humans nowadays is that people eat too much cholesterol. We actually get more than what we need, and then it can build up in the bloodstream and cause problems with heart disease because it can clog blood vessels. But you know, overall, the cholesterol does have an important function. So too little cholesterol in the diet could also be a problem. And then, second function of cholesterol would be that it is a component of the cell membrane. So the cell membrane is not just phospholipids. It also has some cholesterol interspersed. It's mostly phospholipids and proteins and things like that, but there's some cholesterol molecules within the membrane, and it helps stabilize the membrane. And I'll show you a little bit more about that. So let's say we have a cell membrane. We're just going to draw it real simply like this, simplified structure. Sort of an abbreviation where the phosphate heads are these round circles, and the fatty acid tails just kind of stick inward. And the membrane, obviously, we know is this bilayer with the hydrophobic fatty acid tails in the middle, and then the hydrophilic phosphate heads on the outside. Well, sometimes what happens is cholesterol molecules will also be found in here, and they are integrated. They almost like pretend to be a phospholipid. They kind of just blend in here. So I'll just draw the 4-ring structure. I'm going to simplify it because I'm going to leave all the extra chains and all that stuff off. So one of the rings is a pentagon, the other 3 rings are hexagons, and then there's this chain coming off of here. All right, so this part of cholesterol is really hydrophobic, and then the OH up here is hydrophilic because it has that slight charge, so that likes the water. And then you have more phospholipids just sort of blending together here like that. You might have some cholesterols down here also. I'm just going to draw the one in because it does take quite a while to draw. Oopsie. All right, so what does cholesterol do for the membrane? Well, it stabilizes the membrane. So at high temperatures, when the membrane tends to get too fluid, because it tends to get more liquidy, it keeps the membrane from falling apart. It prevents it from disintegrating. So at high temperatures, it stabilizes. and prevents it from falling apart, essentially. And then at low temperatures, it does the exact opposite. At low temperatures, that membrane tends to freeze solid because it's too cold, and substances will turn into solids, or tend to, as the temperatures go down. So the cholesterol molecules sort of interfere with the lining up of the phospholipids, and they prevent it from solidifying. So we have stabilization at both temperature extremes. So cholesterol is an important component of the membrane that stabilizes the integrity of the membrane, prevents it from falling apart at high temperatures, and prevents it from from freezing, at low temperatures. All right, so I also mentioned that cholesterol forms steroids. So here's just two examples of steroids, like testosterone and estradiol, which is also known as estrogen. So estradiol is just the main type of estrogen. Estrogen is like a class of steroids, estrogens, I should say. So estradiol is the most common one. And you can see how they're very, very similar to cholesterol. There's just some slight modifications. And you can also see that estrogen and testosterone are very similar. There's some minor differences over here. So cholesterol is important to make these for regulating the human reproductive system, or animal reproductive system. It's not just humans. All right, waxes we're not really going to worry about. They have a strange structure where it's like basically a fatty acid that has another carbon chain attached to the other end. And yes, they're found in earwax on a human, and they're found in plants to coat the leaves to make them waterproof. Obviously, waxes are important for animals like bees and things like that to build their the honeycomb. But we're not going to worry too much about waxes because they're kind of a minor lipid component. All right, we'll end here for that.